edition of the Iowa Pork Industry Center's webinar series. This month, we're very pleased to have with us Drs. Guilherme Cezar and Drs. Giovani Trevisan to talk to us about the Swine Disease Reporting System, a new tool for producers to monitor animal health across the United States. Dr. Cezar, he received his DVM and Master's in Veterinary Science from the Federal University of Pernambuco in Brazil. After practicing for a couple of years as a technical service veterinarian for an autogenous vaccine company, Dr. Cezar joined Iowa State University, where he is currently pursuing a PhD in population sciences and animal health. Dr. Trevisan earned his DVM degree from Santa Catarina University in Brazil in 2009. He followed that with an MBA received in Brazil in 2013. He then practiced for almost 8 years before joining Iowa State University in 2017, where he obtained a PhD in population sciences in 2020. Currently, Dr. Trevisan is an assistant professor at the Veterinary Diagnostic Lab and the Veterinary Diagnostic and Production Animal Medicine Department here at Iowa State University, where he conducts work with emphasis on veterinary diagnostic data and field-applied epidemiological research to generate information to support stakeholders to make decisions. And Giovani really does a lot of really fantastic and neat work, certainly with diagnostic data, but as well as taking all this complex information that we get with whole genome sequencing on PRRS viruses in particular and making some sense along with that. But today, these two gentlemen are going to talk to us about the Swine Disease Reporting System, a tool that producers can use now to help monitor health across states and across the nation. So with that, gentlemen, I'll turn it over to you.
Thank you, Chris, for the introduction there about ourselves and SDRS. We are here as a messages of the project. I am, along with Dr. Daniel Linhares, part of the PIs of this project, so it's a team effort there. And I'm just going to briefly introduce the project, and then Guilherme is going to provide the updates about what is the current scenario activity of different pathogens that we have in the industry nowadays. So Chris, can you tell me if you can see our screen?
Looks good, guys. Go ahead.
Perfect. So the agenda that we have here today is going to go over about what is the SDRS, who participates in this project, then present to you what are the tools available and what information is there available, and what is the highlights from the pathogens that we are having the current activity in the industry. So the goal of the project is to aggregate information from different VDLs, but share this information of the activity of different pathogens that are affecting the US swine population. We work with endemic disease, and the information is devoted to help and assist veterinarians and producers in making animal health decisions for disease prevention, detection, management, and control there. So a brief overview of the project there was back in 2018 when the first report was released that contained data for PRRS and Iowa State Veterinary Diagnostic Laboratory only. At that time, together with Dr. Daniel, we were building this project. It was laying the foundation there. And later on was the incorporation of other labs that today we have Iowa, Minnesota, South Dakota, Kansas, Ohio, and Purdue that are part of this project. So it's a consortium of 6 different labs that provides the information and that we share with the industry the results there. The project has been largely funded by the Swine Health Information Center since its conception back in 2017, and continues to receive support from SHIC, and we also have support from USDA, all through competitive grants there that we have for the project. So at our overall level there, what happens is we have these 6 VDLs that share the data with us. We do collect the data about testing and testing results for different endemic pathogens like PRRS, Mycoplasma pneumoniae, influenza, the enteric coronavirus that are TG, PD, Delta, and also for PCV2 and PCV3. All these labs also share with us PRRS virus sequencing information of DOR5, and we do organize that information based on the data that we got, plus feedback from our advisory group we do incorporate that information in monthly PDF reports, audio, video reports that are distributed for the general audience. So if you are not receiving those reports today, you can sign up. The report is distributed freely for the audience. Since we have been able to get this through sponsorship from the grants that are from SHIC and USDA. One additional piece of information that goes beyond those pathogen detection is what we call confirmed tissue diagnosis. So when a producer or vet submits samples for disease diagnosis on tissue cases at Ohio State University, that piece of the report track disease occurrence, and that goes beyond these 9 pathogens that I mentioned. It covers all the diseases that occur in swine population that are diagnosed at ICU-VDL only there. One important component that is we have the data that comes from the labs, but we also have the information that is brought by the advisory group to the project. The advisory group had a set of producers and veterinarians that they are distributed across the US, but they provide input of what they are seeing with the boots on the ground there, and also they guide us toward the direction that the project should go in the future. So thank you for all of them for providing this support for the project. And just to have idea how we distributed information, we do have web content that goes on the LinkedIn. That is one of the platforms that we put the reports. There are a video report that's compiled every month that goes through YouTube, but there is also the audio report from that video that is what we call the podcast. And that goes for different platforms that you can access that, download and listen when you are driving across farms. So you can get a snapshot of the updates on the report of the detection, and also a panel of, there is a specialist that comes on board and provide that information every month about different subjects. The report has been distributed by email, so if you want to receive that, you can sign up, and Guilherme has a QR code at the end for that, and you can get that report pushed to you every month when there is a new release. We have about 400 receivers there, and that goes through different countries, not only US, that looks into information, but it's available different channels, and that's information that's out there for you that can be used for your decision making. So I'm going to pass over for Guilherme, that's going to provide you the fun facts about what's going on in the field and how is the activity of the different pathogens that we have there in the report.
Okay, thank you, Giovani, and thank you for the IPIC for the invitation to speak about the SDRS today. And we are going to go through the order of the report. The first page of the report, when you open the PDF file, we talk about PRRS virus. So I'm trying to go to the updates that we have for 2024 and what's going on currently right now in the month of November. And just to start, that's the first chart that you're gonna see in the SDRS report, that this is the total number of cases that was tested by PCR for PRRS in the 6 VDLs that Giovani mentioned that we have in the project. So when you look to the y-axis, that's what you have, like the number of cases. And in the x-axis, you see the months of the year. And then when you look to the columns, you see the number of negative and positive cases based on the color. So when you see this green color, is the number of negative cases, and when you see the red color, is the number of positive cases. And I just would like to call your attention by the month of October of 2024. So last month, you can see that we have a spike in the number of PRRS virus cases that were tested in the VDL. And you can see that the red color is increasing over there. So the number of positive cases of PRRS is increasing in this previous month. And just for you to have an idea, a case not necessarily has just one sample. So for example, one submission can have 20 samples inside of the submission. And then if you have at least one positive sample, then we consider this case as a positive case. And just for you to have an idea of this positivity of PRRS virus, in this chart right now, you can see the percentage of positive submissions by age category. So in the y-axis, you can see the percentage of positive, and each line represents one age category. So when you look to the red line, this is the positivity of submissions coming from the wing to finishing sites, so growing animals, nursery sites, and you can see that for PRRS, it is spiking up right now. So the month of November right now, we updated this chart for you guys. You can see that it's coming up above the 50%. So a lot of submissions for the wing to finishing sites coming positive for PRRS. And also you can see that the cell farm is not increasing too much yet, But for the previous years of the SDRS, we see that when we have this spike in the win to finish sites, on the following month or even the same month, we start to have the increase in self-farm going on as well. So it's something that we want to call your attention. And in the overall submissions, there is the black line that you can see in the chart. You can see that is increasing as well, mainly driven by the win to finishing sites, because this line, we are putting all the submissions together and see how many of them are coming positive, in this case for PRRS virus. And then just to show you the idea of how PRRS virus is above the expected in terms of positivity, in this chart right now, you see the weekly number of positive cases for PRRS, and this is a forecast chart. So when you look to the blue line in this chart, you see what is predicted for the year of 2024. And this blue shade that you see around this blue line is the 95% confidence interval. So what does mean? Is that if this red line that you see in the chart, that is the actual percentage of false submissions going on a weekly basis, if this line go above this blue shade, it means that based on the historical data, we are having more PRRS than in the past. So you can see in the previous week, the week of 43, that was 3 weeks ago, we had this spike in PRRS that is going on above the expected when we look to 4 years behind in terms of historical data. So it's some alert that we are trying to make for you guys that we are having more PRRS than in the past years. And then just to even emphasize what's going on in terms of more activity and above the expected, in this chart right now, we plot more years of data. So you can see that goes until 2020, and each line represents one age category again. So the red line is the win to finishing sites, and the blue line is the self-farm. And when you look to the win to finish sites, you can see that, again, as I mentioned before, it went above 50% of the post submissions. But when you draw a straight line, you can see that this is above or of all the previous years until 2020. And we plot all this data to see how far it goes above of all the historical data. And the last time that we get this sort of positivity was in November of 2018. And besides that, all the other months of the previous years, we didn't have this spike of positivity of PRRS that we are having in the win to finish sites right now. And then for give you more information where these positive cases are coming from, the majority of them are coming in this order. So Iowa predominantly, but then Minnesota, Oklahoma, and Indiana are having more of these positive cases in finishing sites for PRRS in the month of November. And then another good information, we were talking about the PDF chart of the report, but we also have the online dashboards that you can go, and this is real-time data, so every day we updated this information, and you can go there and access for free. If you scan the QR code here on the screen, you go to the SDRS website, and then you can click in the dashboards. And the information there is even better for you, producer or veterinarian, to take a look in your region, the region that you work. So as you can see here in the dashboard, we flag PRRS virus in the month of November, and I will try to explain you here with the laser pointer how the color code works. So this is the overall cases of PRRS virus, so submissions in the VDL. And what we do here, we plot the historical data and compare the historical data of what's going on right now in the month of November to see which states are having activity above the expected. So in the color code, when you see one state with this dark red color, it's because we are having activity above the expected for these states compared with the historical baseline. So you can see that South Dakota, Iowa, Indiana, and Oklahoma are having a lot of PRRS virus activity. When you see in this map, the green color is because these states are having activity within the expected. So what is expected for Minnesota, for example, is going on right now in terms of positive submissions. And when you see this dark blue color here, it represents states that are having activity below the expected when we compare with the historical baseline. So this information is there online for you to access, and you can access the information in an overall level, but you can also see in the win-to market and also adult self-farm in the same dashboard. And you can flag each of these pathogens here so you can get information about all of them based on what you want to see. And also we have in the dashboards online, this is PRRS virus again, But you can look to the sequencing level, as Giovanni mentioned, that we have in the SDRS project as well, this information. And these, you can see the main lineages and the main RFLPs that are being detected in the field in a yearly basis. So again, to explain how the dashboard works, I will go here with the laser pointer. So you can see this is online and you can click in whatever type of information you want. So if you want to see the sequences by phase, you can click in cell farms or in winter finishing sites. If you want to flag on a specific RFLP, you can do that as well. And you can even flag only wild type PRRS virus, for example, or one type of vaccine if you want to see the main lineages that you have. And then the information that I want you to extract from here is the L1C.5. You can see the proportion of this lineage, how it's increasing over time. Before, for example, had more L1A that is decreasing, and the proportion of L1C.5 is becoming one of the main lineages detected in the field. And we're going to break down this information further here for you to see the amount of sequences that are coming for this specific lineage. And as I mentioned, just for you to take a look, that's the most predominant lineage that's been detected in the field. And again, for those who are not familiar with the new nomenclature, this is the old L1C variant that was a very aggressive strain that caused a lot of clinical signs in the field and a lot of mortality. And when you look here to the data, look at the proportion. So this is the year of 2024, and we have almost more than three times the number of L1C.5 detected compared with the second one, that is the L1A RFLP 174. So this is the top 5 that have been detected in the US in 2024. And you can see that the L1C.5 is reaching almost 2,000 sequences. And this represents 50% of all the wild type PRRS virus detected in the field. And another lineage that I would like to emphasize here is the L1C.2 RFLP 124. This is a lineage that was not in the top 5 of the sequences detected never before in the SDRS. And right now you can see that is the third one most detected. And the advisory group that we have in the project advised us that this lineage is having as severe clinical signs as the L1C.5. So it's also a concern that we have in the field. And just for you to have an idea, this information we have in the PDF report as well, you can look to your specific region and see which lineage is more predominant there. And when we go to the L1C.2 that I was talking before, you can see that this lineage is the third most detected in Iowa, and is the one most detected in Illinois. So you can have this information and see why it's spread so much and which states are suffering more with this specific lineage. But if you look to this kind of reddish orange color for the L1C.5, you can see that, again, is the predominant strain in multiple states like Iowa, Minnesota, Missouri, and also South Dakota. So it's one of the main concerns that we have for the swine industry nowadays regarding to PRRS. And then another capability that the SDRS have is to plot this data by state, so you can see which states are detecting this specific lineage and the amount of sequences that were detected. And then in this chart here, you can see this map, and you have these pie charts in each state. And when you see that the circle is increasing, it's because we have more sequences coming from this specific state. In the example, you can see the Iowa here. And the color code represents the type of farm that is coming from. So you can see that the red color represents the win to market, the win to finish sites. And this is for the lineage L1C.2 that I mentioned before, that was predominantly detected in Iowa and in Illinois, but you can see that several states already detected this specific lineage. And you can also see the changes in the dynamic of detection. You can see that in Iowa, the majority of the sequences are coming from finishing sites. But when you go to the Illinois that is suffering with this lineage as well, you can see that the majority of them comes from the South Farm when you see the blue color predominantly in this specific circle for Illinois. And then, as I mentioned, the L1C.5, that was a concern, the old L1C variant that has severe clinical signs. You can see the difference of the amount of sequence that are coming from this specific lineage. And look at Iowa, that is almost 1,500 sequences coming from the L1C.5. And then, but you can see that Minnesota and Missouri also have a high amount of this specific lineage. And Nebraska as well is an example of these lineages start to be detected there with only few cases. And then right now is reaching over 100, the number of sequences coming there with this specific lineage. So once it hits a state, it has a chance of being endemic like this pretty fast. And one information that I would like to emphasize is for South Carolina that never had the detection of this strain before. This was at the beginning of the year, so the good news is that we had this detection. Somehow it was eliminated from the state and we didn't have any detection anymore. But it was a concern because North Carolina as well never had a detection of the L1C.5. And since it's really close, South Carolina and North Carolina, it was a concern for one of the largest breeding herd inventories that we have in the US. But again, another good information is when you look, for example, to Kansas in Oklahoma, this is for 2024, and you can see that they didn't have a single detection of this lineage. So some of these states are being able to keep this lineage out once they have a detection because they had before, when we look to our database, they were able to eliminate and not let them become endemic in this specific region. So still within PRRS virus, but right now we are moving to another tool that is available for you, veterinarians and producers, to use in the SDRS website. And this is the BLAST tool. That this you can compare your sequence that you have the result of one of these VDLs to the whole SDRS database to look for similarities between your sequence and the sequences that we have in our database. And just to remind you, the SDRS has over 110,000 sequences stored there. So it's probably one of the largest database for PRRS virus sequences. And then how it works. That was work made by Sirijita. She's a graduate student here at Iowa State. And this is already online and you can use it. That's the layout that you're going to see when you open the page. And then you can simply copy and paste your result of the VDL of your OR5 sequence. And then just click on the blast button here. As you can see an example, you're going to see the sequence and all these letters that are the nucleotides that you copy and paste. And then you are able to get these results out of the BLAST too. So you're going to see the degree of similarity of your sequence with one SDRS sequence in the database and which lineage and RFLP were, the state that came from, and the received data as well. So you can use this tool as a epidemiological tool for you to take a look. Okay, I broke with PRRS virus and it was this specific lineage. I mean, which state have a similar sequence as mine, for example? And then you can trigger some investigation of how this specific strain came to your farm. But then what I would like to advise, once you do that using this database, take a look on the degree of similarity level. When you see this 100% here, it might be that your sequencing is matching with your own sequence in our database. So the advice that we have is that when you do that, use this specific tool, take a look on the received date. If it was the same date that you submit your own sequence, probably this is your sequence in our database. But if it's not, then you can compare and see the degree of similarity that you have in the BLAST tool that is available for us and for you guys. Right now, we finished the PRRS virus part, and we are going to enteric coronavirus, so PED, Delta, and also TGE. And then we're going to start with good news. This is regarding PED, and then what I would like you to show here is the percentage of post submissions for PED, and this is by season that you can see in the x-axis. And this summer of 2024, we reached the lowest detection of PED in sow farms since this virus came here in the US. So this is good news. It seems that we are taking better care of this virus to get out of our breeding herds, and then we have lowest positivity in the summer. We always have lower positivity in the summer when we look to the historical database, but this was the lowest one since 2014. But then it's not something that, oh, we can be relaxed about PED. No. When we look to the plot of weekly number of positive cases for this specific virus, you can see that some states are having increased activity. And I would like to make an analogy of this chart with an electrocardiogram, for example, but with a difference, there is an inverted electrocardiogram, right? When you are in a healthcare, if you see a flat line, that's a bad sign, right? It's a sign that you are dead. But if you see like these spikes, it's something that you are alive, right? And here we want to see the opposite. When you see the flat line, it seems that this state is having low or zero activity of this specific virus. And when you have the line going up, it's because the activity of PED is going up. And when you look here in the arrows that you have, the red arrows, you can see that Iowa, Minnesota, and Missouri right now had some specific spikes for PED positive cases. And we just updated this chart this week, so this is from the last week already. And then when we look to the other states as well that we monitor in the SDRS, you can see that Oklahoma and Texas are having positive cases of PED as well. So some alert is already starting this activity of this virus. And then we have to increase our biosecurity to keep this virus out of our farms. And then another good information that we have when we plot the historical data in the SDRS, this right now is Delta coronavirus. And this is the number of positive cases per month that you see. And I would like to take a look in these blue arrows first to see that every other year we start to have an increased activity of Delta coronavirus. So when you look to 2020, 2022, and 2024, we have this spike going on in February and March, for example, but it's not as high as the ones that you see here in the chart in 2021 and 2023. And the alert that we want to make is that if we are having this activity above the expected every other year, probably in 2025 right now, in this upcoming winter, we are going to have increased activity of this specific virus. And PED kind of repeat the same pattern, but with the inverted years. So 2020, 2022, and 2024, we had more PED activity, and 2021, 2023, we had less. So it's something that we could see looking to the SDRS database. And then, as we mentioned, we have kind of this window of opportunity in the summer that we have lower activity of dysentery coronavirus. So when you look here in the summer, in the months of mainly June until August, you can see that the activity of these pathogens decrease. And then maybe our industry has this window of opportunity in the summer to try to keep these pathogens out of our farm or try to eliminate it. And just for you to see more clear, this pattern, again, our last chart that we plotted, but as you can see with the blue arrows right now, in the month of August and sometimes in November, you can see very few number of positive cases, around like 5 to 10 positive cases that we have in this specific time of the year. And when we plot this information only for sow farms, so we take a look for a breeding herd only, you can see that it's even lower. So you can see that in September, we had fewer cases since 2020. In 2023, for example, was only one positive case coming from a breeding herd. And right now in 2024, in October, we have really few cases as well. And let's see what's going on in November. We didn't finish the month yet, but it seems right now that it's still really low, the detection of Delta. And it's not a matter of testing that you can see in this yellow box here in the slide, that all these months we have at least more than 1,000 cases submitted in these VDLs that was tested for Delta coronavirus, with an average of 3 PCRs performed in these cases, and really few of them were positive, as you can see in this chart. And then that's one of the best news for the US industry, the number of TGE cases. And you can see that since 2021, to be more specific, March 2002 was our last positive case for TGE. And since then, we are not detecting this pathogen anymore. And you can see that's not a matter of testing. Again, we had over 150,000 cases submitted in these VDLs with no positive cases since then. So it seems that is either circulating at a really low prevalence, or we did eliminate this virus from the US without a lot of effort, and we are not seeing any more TGE in our industry. So right now we're going to move to mycoplasma, is another information that we have in our report, and some trends that we are seeing that we are being successful in keep these bacteria, out of our breeding herds. And then I'm going to start to you when you look to the number of cases submitted in the VDLs for mycoplasma. If you look to 2023, for example, you would be concerned of, oh, we start to increase our number of cases for mycoplasma. So people are testing more for this bacteria. So probably we are having more activity in the field. And that's the beauty to have like more information that not only the number of cases, but also break down the specimen that was submitted and also the positivity. So in this chart right now, you can see the main specimens that were submitted in these cases for the 6 VDLs that we have in the SDRS. And you can see that the number of deep tracheal swabs starts to increase in 2023 and 2024. And this is a sample type that is mainly using for surveillance to keep track that your breathing herd is negative, to take a look in your guilts as well, and in terms of exposure when you are trying to eliminate this pathogen for your breathing herd. And then you can see that the proportion increase of testing of deep tracheal swab. And when we look to the positivity, again, this is the blue line, the cell farm, and the red line, the win to finish sites. And you can see that the positivity in the breeding herds are really low compared to the historical data, and it remains low since 2022. So it's a good sign that we are having less positive cases from mycoplasma in the field. But one concern that we still have is the finishing sites that you can see that is spiking up. But again, we are having more activity of mycoplasma in the finishing sites, but we are doing a good job to keep this bacteria out of our breeding herds because we are having lower positivity over there. And then just to emphasize for you that we keep monitoring that. As you can see, the finishing sites are having increased activity of mycoplasma, but the breeding herd is still low. But we always keep track of to see if we are having more clinical cases also, and if these might affect our sow farms in the future with all this increased activity in the finishing sites. And then this is just like the cherry on top of the cake to see like how much is decreasing over time the number of mycoplasma cases. And right now in this plot, what you are seeing is the number of confirmed disease diagnoses only for ISU-VDL. In this specific chart, these cases were analyzed by diagnosticians, so were tissue cases that were submitted in the VDL, and the diagnosticians analyzed lesions in this tissue to see if they have any evidence of mycoplasma in these specific cases. And you can see that 2020, we had over 160 cases in the last quarter of 2020. But when you look to the trend over the years, in 2024, we are having the lowest amount of mycoplasma cases compared with the previous year. So it is pretty good evidence that we are having also less mycoplasma clinical cases going on in the field based on this database that we are showing to you guys. So that's another thing that the US industry is doing well, eliminating mycoplasma and controlling better in our breeding herds. And right now I'm gonna move to more endemic pathogens that we have in our report. So you can take a look what is the picture right now in November. And we're gonna start with influenza A virus that since October, we are having increased activity of this specific virus going on. And right now in November, we had a spike in the sow farm, as you can see in the blue line color that is increasing. And the win to finish remains high as well for influenza. So we are having more activity of influenza A virus in the field right now. And then another surprise is that, I mean, we were having increased activity of PRRS as well, right? And then we start to plot, let's plot these two viruses together to see how they behave in terms of positivity. And it was a surprise for us when we plot that. In this chart here, when you look to the green line, you see the overall, so among all the influenza cases, how many of them were positive in these submissions. And then the blue line, you see the PRRS virus case. And it's really impressive how the lines kind of follow each other. And you can see that right now we are rising the number of PRRS virus and also influenza positive cases going on in November. So an alert for the industry that we are having more of these two pathogens circulating in our farms nowadays, right now in this month of November. And then this is the information that you can get in the report as well, in the PDF report. As I explained, we have the confirmed tissue diagnosis, that this is cases from the diagnostician that they analyze the tissue to see confirmed evidence of a pathogen causing this lesion. Not a surprise, this is for November as well. We just plotted that. And you can see that PRRS is the main pathogen detected in the ISU-VDL causing lesions in these submissions of tissue, followed by other, this is the top 10 pathogens that we have in the PDF. And another information that I would like to emphasize for you is the Mycoplasma hyohyaenus that you can see on the last position here in the kind of orange color. And this pathogen has been in the top 10 since September. And that's a surprise for us because we don't see that much Mycoplasma hyoharnis being on the top 10 consistently over time. And we are seeing since September in 2024. So it's also a concern in the field that I would like to emphasize for you guys. And just to help you how to interpret the charts that we have in the SDRS report in this part of confirmed tissue diagnosis, again, I will take advantage of the laser to show you. This is the number of cases in November for the nervous system that affected only the nervous system. And when you look to this bubble chart here, there is an Euler diagram. You can take a look of how many cases we have for each pathogen or that have more than one pathogen involved. So just for you to have an example, the Streptococcus suis, for example, so far we had 20 cases that had only Streptococcus suis diagnosed in the VDL, in the ISU VDL. But then when you look to the red bubble here, the red bubble is inside the blue bubble as well, which represents that we are having 2 cases that had Streptococcus suis and also Actinobacillus suis in a nervous system case. And that's how you interpret this chart that we have in the SDRS. And I chose the nervous system first for you to take a look and be easier to understand, because take a look on the respiratory chart, how it's more complex, how we have more diseases affecting the respiratory system of the swine. So again, you can see that PRRS virus is predominant. The red color here in the bubble, you can see a lot of cases with PRRS virus being involved, only PRRS virus being involved in cases in 132 times. But you can see how many they have interactions with other several pathogens. So Streptococcus suis, Pasteurella, Glasarella. So you can see how the respiratory complex involves more pathogens affecting the lesions that we see in the lung mainly. And then the alerts that we have in this chart here is the weekly number of confirmed tissue diagnoses from the ISU VDL. And when we see the columns reaching this specific red line that you see in the chart, it generates one alert that you see this red arrow. So this represents that the number of cases are being above the expected for this specific week. And right now you can see in your screen the Pasteurella cases, how many consecutive red arrows you will see since October right now and the first week of November. So it's spiking up right now, the number of Pasteurella cases. But not only Pasteurella, you can see the Streptococcus suis also having consecutive alarms, influenza having multiple alarms as well, an increased number of cases, and PRRS virus also having consecutive alarms. So activity of endemic pathogens really high in the field right now, and we need to be concerned about all of these pathogens circulating in November right now. So that's how we end the presentation. And if you like the information of the report and everything that we presented here, you can just scan this QR code and we're gonna sign up. You're gonna sign up to receive the monthly reports. And through the report, you can access all this information of the dashboard, the blast tool. So it's super easy. And as our colleague, Dr. Schneider, told us once, that's really easy information for you to take a look. Like one day, 5 minutes of your day, you can scan the whole report and see how the activity is going on in the field for these multiple pathogens. So it just not take a lot of time from you, and you can be informed of what's going on in the swine industry in a monthly basis. So thank you all.
So Guilherme, with all that information that is shared out there, the report is easy to scan and go there. So if your vet are not familiar with that, you can ask for, hey, what are you seeing in the SDRS report? What is the information coming out there? You can compare your system with the SDRS. And we know there was a lot of activity, a lot of different pathogens that are very high activity right now. So is the time that we may reinforce and revisit with our caretakers about, hey, biosecurity practices when you are moving across site, potentially take those measures, change your cover boots on your shoes when you are walking. If you have a bench system, change your shoes, change your clothes to avoid carrying things from one side to the other. But also the biocontainment, that is also the approach of not leaving the pathogen, if you have a positive site, to go to another one. So if you share labor across sites, try to use site-specific clothes to, if they can have a downtime between sites, that's important. But it's important now to revisit those biosecurity practices, what's been done, to try to avoid the larger spread of pathogens. Usually it's a period of the year that Thanksgiving week, we see more activity. Looks like there is a more breaks in biosecurity. So we are in the time of the moment to revisit that and keep vigilant to try to reduce as much as possible this dissemination of pathogens across sites, and also how you care the animals, trying to provide a good environment. There is a lot of changing weather right now in more in the north region of the US. So keep the environment of the animals in a good condition is very helpful too.
All right. Thank you very much, gentlemen. Got a couple of questions here, and just for certainly the live attendees, if you have any questions, you want to go ahead and put them in the chat or unmute yourself, you're welcome to. I'll got a couple of questions just to start off the discussion period. One would be, obviously we've seen the spike here recently in week 42 or 43, and I know you guys have an advisory board that you occasionally get some feedback from. Were you able to assess or get any feedback for potential reasons why we saw that increase so quickly in PRRS positive cases just recently here in weeks 42 or 43?
Yeah, Chris, it's still going on, this information with the advisory, just circulate this high positivity that we have with them this month. And there are different opinions already, like some of the advisors share with us that they were doing more surveillance for this specific finishing site. So sow farms that are already broken, they were just confirming these specific animals to see what's going on. So it's not new introductions. But some of them share with us that they were having more PRRS virus activity in the field, what they saw. So right now that is still circulating with them. We don't have like a lot of information about that. But so far, what they share was that like some of them having more surveillance based on sow farms that broke before, and some of them had lateral introductions in the finishing sites, and that's why they saw this spike that was as high as we saw in 50%.
Another two things that we can see there, Chris, based on the data of the CRS, is the activity of PRRS virus has been higher this year in the finishing sites than the previous years. Usually in July and August, we see a more deeper decrease in detection. We did not saw that very significant this year, so it kept a lot of activity going on there compared to the previous year. So looks like we have been having this activity for a longer time, and now is spiking pretty fast during these months. Additional to that, there is really the sequencing pick up a lot in October, so that means there is a need for more diagnosis, more activity in the field. But one specific virus, that's the lineage 1C.5, is having lots and lots of detection in Iowa. We know that virus has ability to replicate and produce more viral load concentration in sample, more viral particles. So looks like the pressure of infection at regional levels also increase, and that contributes for these outbreaks. We saw that in the finishing sites, but also if you look for sow farms, the activity is not really going up as much there. But if you look for other project, that is the more swine health monitoring project that monitors outbreak in breeding herds, they start to have quite a significant number of outbreaks there that's going on above what we expected for this time of the year. So that's more when you merge two data, you can see that there is lots of PRRS virus occurring in the field right now.
Yeah, very good. Appreciate that. Yep, that does make sense. Like you say, certainly seasonally, and with the virus that tends to put more virus in the environment, and certainly as it gets into the cooler and damper season of the year too, we know that it can persist longer outside the host as well.
Yeah.
So
and the other thing that goes on this time of the year is for some regions you have the manure pumping, so
Yep.
you agitate that virus in the pits, and we have some studies that was done by Dr. Linhares' team that you can detect virus in the pit, and you have more activity after you do that process, either by agitating the virus, the spill over across the farms, or the pumping of those materials. So that also helps to ignite the process.
Yeah, that makes a lot of sense. Giovanni, question for you, as people start to use whole genome sequencing as another tool to maybe look for recombinations or multiple PRRS viruses involved in an infection, do you see a point, maybe sometime in the future, where that sort of information will be common enough across labs and the output would be similar enough that you would see that sort of information being added onto the SDRS report?
I am very optimistic to say yes. I expect that we can move that to that level and start to use that information for our based analysis and decisions there. We know R5 provides a lot of good information there, but in a couple of specific scenarios where you do R5 sequence and the piglets are telling, hey, we're not behaving like if it was the virus that you expected, whole genome would be useful for that to try to sort out what is going on outside of that genome region. Science R5 represents 4% of the genome and goes, if you do the whole genome, you get everything there. So if it's successful, of course, but in having the ability to do that in the VDLs today, we need to do a little bit more improvement in the tools to have a faster turnaround. And today is about 2 to 3 weeks to get a report back, and also decrease a little bit more the cost, so it's more accessible. Once you reach that stage, I am confident that it's going to have more usage and that the labs will be able to provide that information for the project that can receive that and compile in similar way that we have with the R5 and provide that additional information for the producers and veterinarians.
Yeah, very good. I know we have some more detailed information that comes from Iowa State in terms of some of the other pathogens. So you guys did a very good job of covering that. I'm wondering about E. coli. That's one that, tends to be more commonly seen, particularly in growing pigs. And with that, we have a lot of information about different genotypes and different virotypes, depending on, the fimbrial types and the toxin profiles that we can test for, at least the genes that those bacteria have. Do you see an eventual incorporation of at least maybe that specific information across the different labs in SDRS at some point in the future?
Spoiler alert, Chris, is coming 2025. So we had a grant that we submit for SHIC and was approved to do that work. So we have 3 labs that the data has been aggregated already. Now we are working with the other 3 participants labs to have that data incorporated, and it's coming. Next year is going to be available there. It's just a matter of time now to finish the work and put that information together to release both on the PDF report and also on the online dashboards that can have more capability of do self-exploiting of the data there.
Right. Fantastic. Well, that's good. That's the questions that I have. Thank you very much for your attendance today. Thank you to our 2 speakers for the excellent information. And once again, that information, as you saw, certainly on the presentation, is publicly available information. You can go to fieldepi.org and go to the SDRS page and get that information if you weren't able to access it during the presentation, or you can reach out to myself or any of the speakers to get access to that fantastic information that's updated at real time from all the major swine veterinary diagnostic labs across the U.S. So thank you very much, and we look forward to you joining us again next month for the next installment of the Iowa Pork Industry Center webinar series. Have a wonderful afternoon.
Thank you, Dr. Rademacher, for the opportunity. And if you don't have my contact or Guilherme's contact, is in the PDF report, along with the contact of Dr. Danny Linhares, you can find there.